Firstly, Tad sends his apologies for not being able to make it. He fell very sick a day ago and asked me to do this on his behalf. And without much further ado, I'm going to start to tell you a little bit about what we do in the lab and a lot more about what I do in the lab, since Tad's not here. So as some of you might be aware, our lab focuses on one of the major factors that influences community assembly, which is the sequence or order of arrival of species and their timing in determining what the community outcomes are going to be. Very simply, if you have a species pool with a number of species, depending on variation in the order of the species migrating to a newer species pool, you can have several different outcomes. And this sort of an effect is called priority effect and is particularly important in the Nectar microbe system, which is what we use to study community dynamics. We have a whole range of microbes that can colonize nectar, and their arrival or entrance into this patch of nutrient is extremely dependent on pollinators, such as a hummingbird, and especially for some groups, such as the yeast that specialize in nectar, they are extremely dependent on hummingbirds, and so their arrival timing and order can really affect the outcome of nectar communities. Nectar system in itself is a very simple community. There's a few specialist bacteria and yeast that inhabit it, but because of the short-lived nature, these organisms have to grow really fast before the next pollinator can bring them out and disperse them into a newer patch, because the flowers don't last very long. So it's a short-lived system, extremely dynamic, and on top of that, the species have to face fierce competition for the resources, all the while trying to live in a very high osmotic pressure environment and with very little nitrogen being available. So what you end up with is a bunch of highly specialized organisms that compete fiercely for this resource. And that basically makes priority effect even stronger in this environment. And I'm going to show a very simple case study of one of the nectar specialists, Candida rhencensis. And what we can do in the lab is simulate this environment by introducing one organism into sterile nectar and then changing and then introducing another organism in a couple of days to simulate this order of arrival, and then change the order of arrival to basically reflect what would happen as an influence of this variation in the order of arrival. And what we see basically on this plot is the growth of a focal species, Candida rhencensis, if it arrives first in the nectar, followed by a whole range of competitor species arriving second in the nectar. And also when we reverse this order, where the competing species over here arrive first and then Candida arrives second. So we're just looking in this plot at the growth of Candida rhencensis. So in the black bars is when Candida arrives first, and what we see is that it does really well, grows without a problem, but when it arrives second, so the order is reversed here, we see dramatic decline in its growth when there are the Metschnikowia species are the competitor, which basically shows very different outcomes if you change the order of arrival of species. So it's a very simple concept, and it's deterministic in the case of nectar environment. So why this is important is because the nectar community composition can really affect the plant host fitness. While some nectar microbes, such as these yeasts that we work with, have no effect, or some other studies have reported a positive effect on the plant fitness, other microbes, such as these bacteria, really deteriorate the quality of the nectar, and leading to basically changes in pollinator behavior such that the host plant then suffers from the lack of pollination. So in a more ecological sense, this is actually an important effect that we observe. But while we know a lot more about priority effect and its ecological significance, we know very little about what features actually mediate, especially what genomic mechanism mediate this priority effect in these communities. And on top of that, we know even less about the evolutionary understanding of the priority effects as how it came to be. So we decided that we wanted to try our hand at genomics and bring in these amazing tools to try and understand what's happening to these organisms. And we decided we were going to work with Metschnikowia reukaufii for several reasons. One of them being that it's the most dominant nectar yeast and is found globally. It also exhibits high natural morphological variation, and we use these very highly technical terms to describe them, such as smooth and bumpy, because that's the kind of morphology they exhibit, grown on rich media. So what's really nice is that this morphological variation actually correlates really well with some of the physiological variation that we've looked at, such that the bumpy form, the bumpy phenotype, grows faster than the smooth phenotype. So this is just population doubling time over here. And it's also correlated with onset of production of chlamydospores, which again is something that we don't understand what the role is, but it's something we've been observing. So this gives us a really nice morphological variation to work with. But what is the ecological significance of this morphological variation? Well, we did some direct competition analysis, similar to the one that I talked about before, where you introduce one organism first, and then you introduce another organism second, and then you see how their growth changes, and then you reverse the order, and you see how then their growth is affected. So we basically grew Metschnikowia reukaufii, the two different morphotypes in the presence of a competitor Candida rhencensis. Similar to the previous plot, we're just looking at the growth of Metschnikowia reukaufii strains here. Smooth strains are on the top panel, bumpy strains are on the bottom panel. And so if we look at the growth of bumpy strains first, we see that it's only affected in the case where Candida rhencensis comes first. So it's not really affected when its growth is pretty good when it's growing on its own, and then Candida rhencensis comes in second. But when we reverse the order of arrival, its growth is negatively affected. However, in the case of the smooth strains, what we see is that its growth is basically affected every time there's a competitor present, regardless if it comes first or second. And in fact, when it comes second, it's not even able to grow. So for the species, this is basically an ecological dead end, because when a pollinator comes 5 days later, it's not going to pick up any Metschnikowia reukaufii cells over here, while bumpy strains still have a chance. So this is quite a dramatic pattern, ecologically speaking. And we wanted to see that, okay, now that we have this nice ecological variation to work with, what does it mean what does it mean from the genetic variation? Was there any genetic variation that we could pull out of here that could explain some of these differences? To look at genetic variation, you need genetic resources. And that's when I started working in the lab, going where we don't even have a genome for this yeast. Usually people confuse when you talk about, oh, I work with yeast genetics. They go, oh, you must have all these amazing tools. Then I say, yeah, I work with non-model yeasts, which have no tools, essentially. So we started basically right at the bottom. We sequenced the genome, and with our collaborators at Carnegie Institution, Thomas Hardig, we assembled and we developed a custom pipeline to assemble the genome to a very high contiguous level. Basically, the genome is currently, as it stands, is in 7 chromosome-sized scaffolds. It's a fairly small genome, 15 megabase pairs, 7 chromosomes, 6,000 predicted genes, which is roughly what you would expect. Completion rate is quite high, and we also have the complete mitochondrial sequence. So now we have some idea of what's in the genome, and we started probing the genome for signatures of adaptation just to get started. We had nothing, now we have something. We start looking at interesting things in the genome, and what we start with is to look for tandem gene duplications, because tandem gene duplications are one really easy to track mechanism by which organisms accumulate interesting evolutionary adaptations. So what we found, which is very interesting, is first, Metschnikowia has a very high rate of tandem gene duplication. In fact, one of the highest rates reported in the literature. Only slightly better than, for example, another yeast, which is a marine yeast, which is a strange case in itself, at almost 5% of the genome, of the predicted genes showing tandem duplications. And a lot of these genes are involved in nitrogen metabolism and transport, which makes sense because nectar itself is a very low nitrogen environment. What I would like to show here is basically an example of what kind of genes we found. And one comparison here is for a general amino acid permease. And all these green copies are basically the tandem duplicated copies in Metschnikowia reukaufii compared to its closest relative, Candida lusitani, and compared to Saccharomyces cerevisiae, which only has one of these genes. Now, why general amino acid permease is interesting to us is because it's a high affinity scavenger of amino acids. In fact, it's like an open portal for all amino acids that you could get, and it's basically the only one that picks up all amino acids. In most yeasts, it's turned off when, most times when nitrogen is available, and is only active when nitrogen starvation is induced. In Metschnikowia reukaufii, it has 4 copies of this gene, which basically says that this shows that this is a kind of an adaptation possibly to the low nitrogen availability. And another thing that's very peculiar about Metschnikowia reukaufii is that as a nectar specialist, it's able to reduce the amount of nitrogen, amount of amino acids particularly, by more than 90% in less than 2 days, which is pretty cool because when you think about the priority effect that it has on other species, it's in the first few days where the species basically takes over the resource, grows really fast to become highly competitive. And when you have really low nitrogen, and on top of that, something like this comes along and takes away most of that nitrogen, another competitor coming a couple of days later has basically no chance. So we think that maybe we're onto something with a potential mechanism for priority effect, but this is what we basically learned from the genome. I mentioned earlier about the morphotypes that we're working with. So we went ahead and resequenced a whole bunch of strains from the 2 morphotypes. So again, we had 2 different host plants, and we didn't see any host influence on the genetic diversity observed in the genomes of these strains. But what we found was that the there was clear genetic differentiation between the 2 morphotypes, which is nice. We have basically the smooth strains on top here and bumpy strains on the bottom. There's a few in-between strains, but I can talk about them later. But more or less, it looks like that bumpy is a more evolved character. But what we wanted to know was that what these actual genetic differences were, and if we could kind of gauge at where these differences were and what they kind of meant before, so we could select some candidates to go and do some further work. So basically, we developed another pipeline to then mine these differences out. We basically had 3,000 high-quality differences between the 2 strain cohorts. And then we did functional annotation to identify roughly where these differences were and were they associated with any genes of interest. One of the key results from this analysis was that most of the differences were within gene promoters, which is really interesting and which is kind of what you would expect to be a safe strategy rather than losing your genetic architecture. You just modulate how you operate it. And some of the examples of the genes that kept coming up as where these SNPs were located were again associated with these general categories like amino acid transporters or amino acid metabolism, osmotolerance pathway, sugar signaling. So really related to the environment and their metabolism that these organisms are engaged in. So that gives us a lot of clues that these differences between smooth and bumpy are probably due to their metabolic differences and mostly due to regulation, how the way bumpy and smooth regulate their metabolic capabilities. So we thought we'd have a shot at how to see what how they are different in their metabolism, and we did some metabolic screening. We provided them with a metabolic substrate panel, basically including amino acids, organic acids, sugars, and sugar alcohols, and a whole range of sugars. And basically what we see here is each of these rows basically records the response of one strain of Metschnikowia reukaufii, either belonging to the bumpy category or the smooth category, and its growth rate against that substrate. Now, I haven't labeled all the substrate because there are about 100 of them. But what we basically found what is very obvious here is that the bumpy strains are able to utilize a lot more and grow a lot better in under some of these nutrients compared to the smooth strains. In fact, they cluster quite well, and this clustering is basically based on their metabolic response to these substrates. That already tells us that there's something key different between these strains, and that some of these differences that we're finding in their genomes might be able to explain these. Another thing that we wanted to know that, okay, now we're starting to get some handle on what might be happening in the genomes, but are they really, these two strains, are they really ecologically different? In fact, when we go out into the field and then collect these types of strains, do we find any real distributional differences? Because it's one thing to find these differences in the lab. Our interest is more in wide landscape patterns, and we want to be able to confirm this work outside and these effects, that if they have a real impact out in the field. So we went out and did a survey of 12 sites along the San Francisco Bay Area, well, up to Bodega Bay in the north and Big Sur in the south. 12 sites, 96 flora, total of 1,152 samples. And what we basically found was that the bumpy strain was so we basically collected everything that was there. Then we brought them home and we characterized them, well, not to my home, but to the lab, which is kind of like home. And then we characterized all these strains, and what we found was that wherever bumpy was present, it was dominant. So out of the 12 sites, 11 sites had bumpy in them, out of which 7 of the sites, it was dominant, and that in itself is basically, this is a wide screen. We could have had anything, but what we found was that, in fact, bumpy strains is more widespread in the natural landscape. And that kind of gives us even more reason to pursue this question, to really try to understand what is happening and what really causes these large landscape patterns. Further confirming that these metabolic differences and these genetic differences actually play out a bigger role. So the next step is a lot of work for me. First, we're going to do RNA-seq for the Metschnikowia reukaufii to confirm some of these predicted genes that we have. We are also looking at doing some qPCR. In fact, we're in the process of doing this right now, looking at those transporters that I talked about before, and we have some other targets as well to do with osmotolerance pathway and amino acid metabolism. We have some collaborators who are currently trying to get these strains to hybridize, which they don't do naturally, so it's a bit of a tough cookie. And one of the questions that we're really interested in is in why does the smooth exist, because it is still found, and since we keep seeing that bumpy does better, bumpy performs better metabolically, bumpy is more widespread, why do we still keep getting smooth? We have a few theories on this, but I'll leave that for later discussion. And then this is only one of the species. We're really just starting to get at what's happening in the nectar microbial system. We have a whole range of other species that could have similar ways of adapting to the environment, but this is, I think, a good start. And we look forward to doing more of this kind of work with the other species to see how the different species that are interacting at a species level influence this pattern. And with that, I would like to just summarize that Metschnikowia reukaufii strains vary in a way that can tell us something about the genetic mechanisms of the priority effects that we're observing in this system, and that they hold a promise as a model system that we could exploit later as more and more genetic resources are built. And the last point, which lots of work still to do, and with that, I would like to thank some of our collaborators and leave you guys with a very short clip of a hummingbird doing its job. Thank you.
Okay, questions, and we have one right here.
So when you were showing the geographic distribution of the bumpy and smooth, did you actually sample some inland locations as well? Because I noticed it's all coastline, right? So maybe you should ask permission to go to Las Vegas.
Unfortunately for me, because I'm not a big fan of Las Vegas, Mimulus is a coastal plant, more or less. Well, Mimulus aurantiacus, the species of the host plant that we're focusing on to minimize other types of variation that you observe, is a coastal scrub, which is a good thing because you get to go to the beach. So less inland sampling. We tried to get some, but there are a few patches inland where Mimulus does occur, but the seasonal differences mean that when we're out collecting, they are done flowering and are dead already. So this summer, we will try to expand more inland, but last summer, we were basically had our hands full with the beach samples.
Thanks.
Any other questions?
I was curious whether there's variation in the nectar concentration or compositions in the wild, and whether there's variation in the priority effects in the wild.
So that is a very good question, and that is some of the work we're doing currently. We do expect that a lot of this variation that we see is due to these environmental factors. And there is some work in the literature showing that that's the case, that you find different phenotypes sort of attracted to different hosts or different. In our small study that we looked at in the previous summer, we didn't find any host effects, so we kind of laid it off for then. But we're actually bringing that back in at a larger scale now, and we're gonna, so for the samples that I showed at the very end for the landscape level, we plan to do genetic characterization of all these strains, as well as characterization of the nectar that they were sourced from. So those 1,152 samples will have amino acid, sugar composition, and even diversity composition. So that's in the works.
Okay, over here in the back.
Do you have any sense of whether these populations are just completely clonal, or is there evidence for sexual reproduction as well?
So there is a lot of work on that. In fact, that's probably one of the best research things about this particular species. In the wild, they have never been observed to form spores or sexual reproduction having occurred. There is some work from Spain where they found that essentially these are clonal, so you could almost treat them as cryptic subspecies, if you were. But there is some mixture. We don't know where it happens. There are some theories about possibly in the bee guts, kind of like how it works in Saccharomyces cerevisiae, or is kind of like hypothesized that yes, there's a gut enzymes in Drosophila that break down the ascospore, and that's where the sexual reproduction happened. In our case, nobody's really looked very well, but based on the population genetics work, it looks like they are more or less clonal, but they're still very similar, so there has to be some sort of mixing happening.
One more back here.
I was wondering, with your resequencing data, do you find any duplication losses within the strains, between the strains, sorry?
So we haven't actually looked at that yet. That data is kind of on the side at the moment while we're annotating and trying to get the genome out. But yes, that's something we will look at. I would think maybe, but it's likely to be not, because if there are these genes that are present in the organism, I would think that they would stay, like they're still the same species. I think genetic losses would be kind of a major difference. That's my understanding. But what we're finding with our SNPs, which regardless of gene losses, is that they are present kind of in the regulatory regions, and that's probably an important mechanism. But again, we haven't looked thoroughly in the resequencing data yet, but we will be looking at that. Thanks.